It's a very exciting time in the world of Parkinson's research. We have a lot of therapies for symptomatic medicines, but we need more, and we need more targets. So newer types of therapies, and we also need therapies that are focused on the causes of Parkinson's. And what our study does is help us to look and break down additional genetic causes of Parkinson's. And we're looking on multiple levels. We'll be contributing samples to the NIH Parkinson's biomarker program, which we're very excited, so that we can follow up on our research leads, but also other investigators in the US and beyond will be able to look at these samples. And we're contributing samples, and then we're also looking at genetic profiles from people with Parkinson's disease, especially people with LRRK2 mutations, with GBA mutations, and then people without mutations in either of those genes. And we're trying to find additional genes, looking at different types of genes, neurodegenerative genes, genes related to the immune system, and genes related to what we call the lysosomal storage disorder genes. And we're looking at the expression of these genes in monocytes, these particular blood cells that may be very relevant to Parkinson's.
Our project is centered on Ashkenazi Jewish population, which is more of a homogeneous population where we think there'll be theoretically less genetic genes contributing to their disease. And so we're hoping that that will allow us with smaller sample sizes to actually find variants that contribute to risk or to actual disease. But our samples will go into this much larger database and will be, so other researchers can combine them with other studies of Parkinson's disease. And so I think that together it'll be a much stronger study, much more powered study.
Well, I think that one of the things that is so important in clinical trials is that we understand different causes and of Parkinson's, and this helps as far as helping to narrow down different causes, because we hope with Parkinson's that we'll be getting towards a precision medicine approach, a little bit more like a cancer approach, where and it also may not be just one gene. That's the theory, that there are multiple genes that are contributing, and that understanding these different levels will help as far as selection of medicines. So we hope that our work will contribute towards development of new medicines, but also for patients and their physicians to have a profile so that medicines can be properly chosen and selected.
I'm not a clinician, but I do think that, when we talk about Parkinson's disease, it's actually not just one disease, it's many different diseases. And so the idea behind biomarker studies and genetic risk factors and imaging and all of these different techniques that are being applied now to Parkinson's disease is to try to pull apart these different subtypes so that medicine can be directed towards those subtypes.
Yeah, I think that even if people don't have these specific genes that we're talking about, LRRK2 and GBA, that precision medicine will be relevant, that there are other genes, and also understanding even more common variants and how much they contribute. And one of the things that's exciting with LRRK2 and GBA research is the mechanisms that we're understanding for each of these related genes are probably relevant to people without these mutations as well. So it's not just for this group of people with LRRK2 and GBA. It's for a much broader community of people with Parkinson's.